Okay, so welcome back to this next video in which we are discussing endothelium-dependent vasodilatation. Okay, so we've discussed that the endothelium is always sending signals to the smooth muscle cells which surround the arterioles and telling them not to contract and instead to relax. Okay, and we've discussed now one of these principal signals, which is the generation of nitric oxide, which will diffuse to the vascular smooth muscle cells, activate soluble guanylate cyclases, which will generate cyclic GMP, which will then activate protein kinase G, which will inactivate the myosin light chain kinase by phosphorylating it, which prevents it from being activated even in the presence of calcium calmodulin complexes in the cytoplasm, and thereby effectively inhibits the contraction of the vascular smooth muscle cells. Okay, right. So, what we now want to do is look at another mechanism of endothelium-dependent vasodilatation. So going back to this picture here, remember we start off with calcium signals in the endothelial cells that are triggered by shear stress in normal physiology, okay, but in inflammation, certain inflammatory mediators such as histamine or bradykinin acting on G protein coupled receptors which are coupled to the GQ pathway can cause calcium mobilization from the ER in these endothelial cells, which then also causes a calcium signal. So we saw how the calcium binds to calmodulin to form calcium calmodulin complexes, and that this then activates eNOS within these endothelial cells. What we now want to see is another effect of that calcium in the endothelial cells, okay, and this is going to be that it's going to activate the production of prostacyclin, okay. So now what we're going to do is we're going to turn our attention onto prostacyclin, okay, which is also called prostaglandin I2, and for short, prostaglandin I2 is often abbreviated to PGI2 as so. Okay, right, so we want to see how the endothelial cells are going to be activated to produce prostacyclin by this calcium signal within the endothelial cells. Okay, so we start off then with this calcium signal within our endothelial cells. So I'll draw our endothelial cell here, okay, like so. Here is its nucleus, and what we now know is that calcium has gone up inside the cytoplasm of the endothelial cell, so I'll put calcium going up. Right, what this is going to do is it's going to activate an enzyme that is usually located in the cytoplasm of the cells, known as phospholipase A2, or PLA2 for short. So this stands for phospho, that's the P, and then lipase, that's the L, and then specifically phospholipase A2. Now phospholipase A2 is usually in the cytoplasm of the cells, so it's usually just free in the cytoplasm of the cells. However, it has a special domain on it, which is known as a C2 domain, which I'll draw here. I'll colour that C2 domain in a special colour. So this special domain of the phospholipase A2 enzyme, coloured in blue here, is what's known as a C2 domain. And C2 domains are special domains that you find in many different proteins, which are capable of binding to calcium ions. Okay, so when calcium goes up in the cytoplasm, what ends up happening is these phospholipase A2 enzymes end up getting calcium binding to their C2 domains. Okay, and when they get calcium binding to their C2 domains, this causes them to translocate from the cytoplasm to the underside of the phospholipid bilayer, so they relocalize themselves, so that they are now located just underneath the phospholipid bilayer, like I'm drawing now. And now, once they're underneath the phospholipid bilayer, they can set to work breaking down a component of the phospholipid bilayer. So they're going to, just like phospholipase C-beta enzymes, which we saw so long ago now, they too are going to break down a component of the phospholipid bilayer, a membrane phospholipid. But they're going to break down a very different phospholipid to phospholipase C-beta enzymes. Okay, so phospholipase A2 is actually going to break down a much more present phospholipid. So you remember I told you that PIP2, phosphatidylinositol 4,5-bisphosphate, is quite a rare phospholipid in the cell membrane. Okay, phospholipase A2 is going to act on a much more common phospholipid. Okay, and this is the phospholipid phosphatidyl. Okay, so it's got this same prefix, okay, which tells us that the molecule is based on phosphatidic acid, and then it's phosphatidyl choline. Okay, phosphatidyl choline is often abbreviated down to PC for short. And now let me show you the structure of phosphatidyl choline. So once again, I'll show this in the cell membrane. Okay, so once again, its structure is based on phosphatidic acid. So we'll draw phosphatidic acid firstly. Okay, so here, to remind you, are the two long-chain carboxylic acids. Okay, here is the glycerol backbone in green here. And then here is the phosphate group of the alcohol group of the third carbon of the glycerol molecule. And then phosphatidyl choline, what you're going to do is you're going to attach onto this phosphate group, rather than an inositol ring, you're going to attach a molecule called choline, okay, which is the same choline that is used in acetylcholine. Okay, so here it is in blue here. Now phosphatidyl choline is a very present phospholipid within the phospholipid bilayer, and it also has another name which I'm just going to mention. It's also known as lecithin. Okay, so if you ever hear someone referring to lecithin, that's just another name for phosphatidyl choline. Okay, so the phospholipase A2 enzyme has now bound calcium to its C2 domain and has now translocated up to the underside of the phospholipid bilayer, and what it's now going to start doing is breaking down phosphatidyl choline molecules within the phospholipid bilayer. Okay, and what it's going to do is it's going to cut this bond here. It's going to cut the bond between the second long-chain carboxylic acid, or the second fatty acid, and the second alcohol group of the glycerol molecule. Okay, and that's going to produce two separate products here. Okay, both of which are going to remain in the phospholipid bilayer here. Okay, so one of them is what's left of phosphatidyl choline once it has lost that second of the long-chain carboxylic acid. So it still has its first one here. Okay, and there's the glycerol molecule in green there, and then here is the choline molecule in blue here. Okay, right. Now, that molecule is called lyso-phosphatidyl choline, or lyso-PC for short. Okay, and then the other molecule you produce is this free long-chain carboxylic acid. Okay, now, this free long-chain carboxylic acid in some phosphatidyl choline molecules, this will be a long-chain carboxylic acid called arachidonic acid. Okay, and this is the important one, that we are going to start liberating some arachidonic acid. Okay, so this is, let's say that this phosphatidyl choline molecule here just so happened to have a arachidonic acid molecule in position 2 here, and then when we cleave it apart like so, we produce some arachidonic acid as one of the products. And it's this arachidonic acid that is now going to be used by other enzymes to produce prostaglandin I2. Okay, so far you've activated the breakdown of phosphatidyl choline molecules, these essential components of the phospholipid bilayer, into lyso-phosphatidyl choline, and you've also liberated free fatty acids. Some of those free fatty acids will be arachidonic acid molecules, okay, and these are now going to be used as the substrate for producing the prostacyclin molecules. Okay, so arachidonic acid then is our starting point. The first thing that's going to happen is that if we want to synthesize a prostaglandin, arachidonic acid firstly needs to be converted by the cyclooxygenase enzymes. Okay, so firstly it's going to be converted into something known as prostaglandin G2, and then it's going to go on further to be converted into prostaglandin H2. So I'll write these names fully out. So the PG stands for prosta, that's the P, glandin, that's the G, and then this is prostaglandin G2, and then this one's prostaglandin H2. Okay, now, both of these reactions are catalyzed by the same enzyme, okay? And these enzymes are known as cyclooxygenase enzymes, and there are multiple different cyclooxygenase enzymes, the most important being COX-1 and COX-2. Okay, so these are catalyzed by cyclooxygenase enzymes. Okay, so either COX-1 or COX-2 can catalyze this reaction. Okay, right. So these reactions are both catalyzed by the same enzyme, and they overall convert arachidonic acid, therefore, into prostaglandin H2. Now, these reactions are actually so important that they have their own special names. So the conversion of arachidonic acid into prostaglandin G2 is called the cyclooxygenase reaction. Okay, so that's the first reaction catalyzed by the cyclooxygenase enzymes. And then the conversion of prostaglandin G2 into prostaglandin H2 is known as the peroxidase reaction. Okay, right, so that's what's going to happen to the arachidonic acid that we've now liberated from the phosphatidylcholine. It's going to be converted by cyclooxygenase enzymes in the endothelial cell to prostaglandin H2. Okay, the prostaglandin H2 is then going to be converted into prostacyclin, or PGI2, prostaglandin I2, and this is done by the enzyme prostacyclin synthase. Okay, so prostacyclin synthase is going to convert prostaglandin H2 into prostaglandin I2. Okay, the prostaglandin I2 is then going to be released from the endothelial cell, and this is going to go and act on vascular smooth muscle cells. So to stress this really important point once again, these diffusible molecules, such as nitric oxide and prostacyclin, they are not just being produced by the endothelial cells that are actually near the vascular smooth muscle cells, i.e. those of the arteriole. They're also produced by the endothelial cells of the capillaries and the venules in this local area of tissue, and all of these molecules are going to be diffusing back to the vascular smooth muscle cell of that arteriole which supplies this area and controlling its contraction status. And indeed they're going to be trying to make it dilate, basically make it stop contracting. Okay, right. So what we now want to see is how the prostacyclin is actually going to act on the vascular smooth muscle cells, okay, to produce relaxation, okay, and it's going to act through G protein coupled receptors. Okay, so let's have a look at this. So let's say we have our membrane. I'm going to try and draw it in here. Okay, let's say we have our membrane of the vascular smooth muscle cell shown here. So this is the phospholipid bilayer. Let's say this is the outer leaflet, so this is the extracellular fluid here, and this is the cytoplasm here. Okay, so the receptors for prostacyclin are G protein coupled receptors, and they are known as IP receptors. Okay, so I for the I2 here, and the P is for prostacyclin. Oh, sorry, for prostaglandin rather. Prostaglandin I, basically is what it means here. So this is an IP receptor here. Okay, so what's going to happen is the prostacyclin is going to come in and bind to this IP receptor and cause a change in conformation in the IP receptor, and it's then going to be able to interact with a heterotrimeric G protein. Okay, now which heterotrimeric G protein does the IP receptor act on? Well, acts on the heterotrimeric G protein GS. So if I try and draw this in this little space here, what this again means, remember what we discussed last time we were discussing heterotrimeric G proteins. They are named after which alpha subunit you have, not which beta or gamma subunit you have. Okay, so I've made a right mess of this picture, so let me just colour it in to hide the fact. So there's the beta subunit. Okay, here is the gamma subunit here. And now we have the alpha subunit here, which is currently in the off state, and we can tell that it's in the off state because at the moment it's bound to the beta and the gamma subunits. Okay, and here are those lipid molecules off the alpha subunit and the gamma subunit. So here is the guanosine diphosphate, and this alpha subunit is specifically going to be the alpha S subunit now. Okay, and then that's why the entire heterotrimeric G protein is then called a GS heterotrimeric G protein after the alpha subunit. Okay, right, so the G protein coupled receptor is then going to activate the heterotrimeric G protein, causing the dissociation of the alpha subunit from the beta-gamma complex. Okay, so now let's draw our alpha S subunit going off in the membrane on its own. Okay, so here is the alpha S subunit here, which is now in the on state, so it's got guanosine triphosphate, GTP bound to it. Okay, and what it's going to do is it's going to go and activate enzymes called adenylyl cyclase enzymes. So let me now show you the structure of an adenylyl cyclase enzyme. So adenylyl cyclase enzymes are integral membrane proteins. Okay, so they are a single polypeptide rather than being a multi-polypeptide complex. Okay, and they have 2 clusters of 6 membrane-spanning alpha helices. So here is this first cluster of 6 membrane-spanning alpha helices. Then they have an important loop between that first cluster and the second cluster of 6 membrane-spanning alpha helices, which is over here. And then they have an important cytoplasmic domain here as well. Okay, so let's highlight up some important domains of the adenylyl cyclase enzyme. So here, which I'm now highlighting in red, these are the 6 membrane-spanning alpha helices of this first cluster of 6 membrane-spanning alpha helices. And this first cluster is known as the transmembrane domain 1. So T is for trans, okay, then the M is for membrane, and then the D is for domain. So transmembrane domain 1 there. Okay, then the second cluster of 6 membrane-spanning alpha helices, which is over here, that's known as transmembrane domain 2, TMD2. Okay, right. Now, although the transmembrane domains look mightily impressive, they are not the actual active portions of this enzyme. They are the portions that hold it attached to the membrane, but they're not the portions that actually catalyze the production of cyclic AMP. Instead, the portions that actually produce the active catalytic site are actually in these cytoplasmic domains. So this loop between transmembrane domain 1 and transmembrane domain 2, this is what's known as the C1 loop here. Okay, so this is the C1 loop. And this domain after transmembrane domain 2, this is known as the C2 domain. Okay, now C1 and C2 are further divided up into separate domains. So C1 is divided up into C1a, which I'm going to colour in turquoise here. So that's C1a. And then also C1b, which I'll colour in orange here. Okay, so that's C1b there. And C2 likewise is divided into C2a, which is this portion here, and then C2b, which is this portion here. Now I'll colour C2a again in turquoise, and I'll colour C2b in vivid purple. Right, so the reason I've coloured C1a and C2a in the same colour is that those are the portions which are actually going to dimerize together to make the active catalytic domain of the enzyme, i.e. this is an inactive adenylyl cyclase that I've drawn here, and I should have probably put the name of the enzyme at some point. This is adenylyl, or you can call it adenylate cyclase. Okay, in order for the enzyme to actually go into an active state, you need the C1a domain here in turquoise to dimerize with the C2a domain here. So when those two are bound together, that actually makes an active catalytic domain which can catalyze the conversion of ATP into cyclic AMP and pyrophosphate. Okay, so how then does the alpha S subunit, shown here in red, with the GTP bound, actually activate the adenylyl cyclase enzyme here? Well, what it does is it binds to the C1 loop and it binds to the C2 domain here. Oh, and by the way, I probably should say, this C2 domain here is completely different from the sort of C2 domain we were talking about up here in phospholipase A2. They're completely unrelated. This is the C2 domain of an adenylyl cyclase enzyme, nothing to do with the C2 domain that you find in phospholipase A2, so don't let that confuse you. Okay, what has to happen then is, where were we? Oh right, yes, how the alpha S activates the adenylyl cyclase. So it binds to a portion of the C1 loop, it binds to a portion of the C2 domain, and it brings them close together, and that promotes the dimerization of C1a with C2a, and therefore the conversion of this inactive state adenylyl cyclase into an active state adenylyl cyclase. And then once it's activated, what it will do is it will take in adenosine triphosphate, and it will convert it into cyclic adenosine monophosphate and pyrophosphate, PPI. Okay, right, so what's now going to happen is this cyclic AMP that you are generating is going to activate protein kinase A, and maybe you're starting to see where this might go. Last time, the last pathway we looked at, nitric oxide results in the production of cyclic GMP, which results in the activation of protein kinase G, which phosphorylates myosin light chain kinase and stops it from being activated by the calcium calmodulin complexes. We have now produced cyclic AMP. It's going to activate protein kinase A. Maybe you'd like to guess what protein kinase A is going to do. Okay, so let's get another piece of paper and see what it's going to do. Okay, right. So, we've just generated cyclic AMP. Now, let's have a look at protein kinase A. Now we'll see the parallels between protein kinase A and protein kinase G. Now, at first when you see protein kinase A, you'll think it doesn't look anything like protein kinase G, but there are similarities between them. Okay, so let me show you what protein kinase A looks like. Now, protein kinase A is actually a tetramer of 4 different proteins, at least when it's in the inactive state. Okay, and that is the main difference between it and protein kinase G. You'll remember when we discussed protein kinase G that I told you about how protein kinase G could dimerize with other protein kinase Gs. Now, we didn't actually draw this, but in reality, imagine there's a mirror there. You could have another one here, dimerized with this one. Okay, so protein kinase G exists as a dimer, but protein kinase A actually exists as a tetramer when it's inactive. And I hope, apart from that difference there, you might be able to see the similarities between protein kinase A and protein kinase G. Okay, right. So this is the inactive protein kinase A that we're about to draw here. Okay, now the inactive protein kinase A, pKa, is often referred to as a hollow enzyme. Okay, now what that means is that it's made up of multiple different subunits. So whenever you hear someone talking about a hollow enzyme, it means that it's built out of multiple different protein subunits. Okay, so what we are drawing is the inactive protein kinase A hollow enzyme. Okay, now, I've drawn 2 proteins so far, and these 2 proteins are the regulatory subunits. So 2 regulatory subunits dimerized together. So these are the regulatory subunits of protein kinase A. So there are 2 separate proteins there. So regulatory subunits of protein kinase A, pKa. Okay, and then there's going to be 2 additional subunits attached to these, which are the actual catalytic subunits. Okay, which I'll draw here. So these are the catalytic subunits of protein kinase A, whilst these 2 are the regulatory subunits. So I'll write out catalytic subunit of protein kinase A. Okay, now, you will notice that the regulatory subunits, each of them have got these little binding sites that I've shown here. Okay, and that's because those are the binding sites for cyclic AMP. Okay, so hopefully you're now starting to potentially see the analogue between protein kinase A and protein kinase G. Protein kinase G also had 2 binding sites, which were for cyclic GMP. Okay, now the difference between protein kinase A and protein kinase G effectively is that in protein kinase A, the regulatory subunit and the catalytic subunit are actually separate proteins, whereas in the case of protein kinase G, they're all one protein. Okay, now if you imagine that the regulatory subunit and the catalytic subunit were one protein, then this would just be a dimer and it would be exactly like protein kinase G. So you can think of protein kinase A as just being like protein kinase G, except that the catalytic subunit here in blue is actually a separate protein to the regulatory subunit. So there's the catalytic subunit in blue, and here's the regulatory subunits in red. Okay, so this is the inactive enzyme at present. Okay, there's no cyclic AMP bound to those regulatory subunits, and so the regulatory subunits are bound to catalytic subunits, and they're inhibiting these catalytic subunits. So these are not active, they're not phosphorylating anything at the moment. Okay, however, what can happen is when cyclic AMP is generated, 4 cyclic AMP molecules can bind to these cyclic AMP binding sites, that causes a conformational change in these regulatory subunits, and they release the catalytic subunits. Okay, so the catalytic subunits break away from the regulatory subunits. The two regulatory subunits remain dimerized together, but they release the catalytic subunits, and those catalytic subunits are now no longer inhibited by the regulatory subunits, and therefore they're free to go off and phosphorylate things. And what they do is they phosphorylate myosin light chain kinase, which I'll draw here. Okay, so this is going to once again represent myosin light chain kinase here. And what the catalytic subunits of protein kinase A do is they phosphorylate the myosin light chain kinase, just like the protein kinase G enzymes did. And again, this phosphorylation of the myosin light chain kinase prevents the myosin light chain kinase from being activated even if calcium calmodulin complexes are present in the cytoplasm of the smooth muscle cell. And therefore, again, the activation of these IP receptors by prostacyclin on the surface of the smooth muscle cells is going to stop the smooth muscle cell from contracting even if there's the calcium signal present. Okay, so again, this is another relaxation signal coming from the endothelial cells. Okay, right, so we'll call it there for this video, and in the next video we're going to discuss the final way that endothelial cells send signals to the smooth muscle cells to relax. And this is through a phenomenon known as endothelium-dependent hyperpolarization.